Building on years of industry experience, ZeaChem has developed an innovative, economical process for the conversion of renewable biomass into sustainable fuels and chemicals. Based on successful lab results, the pilot facility in Colorado was conceived and constructed at the end of 2009 to provide scale-up of ZeaChem's core technology, specifically the fermentation of sugars to dilute acetic acid. At the pilot scale, ZeaChem has achieved industrial-level fermentation results 10,000 times greater than standard lab scale. The results have demonstrated successful fermentation of greater than 50 grams of acetic acid per liter in less than 100 hours, and the results have been repeated and verified. With the pilot equipment, ZeaChem has been able to acquire sufficient data to successfully design fermentation, filtration, acidification, and microfiltration to commercial scales. ZeaChem selected Morella thermoacetica, a non-genetically modified bacteria, as the fermentation organism of choice for the C2 platform, or 2 carbon atoms. This organism is the basis of production for multiple saleable products, including acetic acid, ethyl acetate, and ethanol. The naturally occurring bacteria is a suitable choice for use in commercial production due to its ability to operate at a temperature of 136 degrees Fahrenheit, an environment in which few other bacteria survive. During fermentation, agitation is used to maintain optimum conditions, and pH is closely monitored. The ZeaChem fermentation process starts with as little as 2 millimeters of frozen cell culture. Successive batches of increasing volumes of bacteria are grown up in order to introduce live cells into progressively larger volumes of fermentation media. In the ZeaChem system, it was found most effective to observe a 10-time scale-up factor. This means that over the course of about a week, we can scale up from a 2-millimeter frozen culture to about 12,000 liters in Seed Tank 3. The seed train, composed of 3 seed fermenters, Seed Tanks 1, 2, and 3, are used to transition from lab-scale to production-scale fermentation during startup, or to maintain live culture between production-scale runs during normal operation. Because ZeaChem uses known processes and technology, most of the equipment for the pilot plant was purchased from the used equipment arena. After scaling up the fermentation through the seed train, ZeaChem transfers the fermentation broth to the broth clarifier. The harvested broth contains about 6.5% calcium acetate and is passed through a membrane in a process referred to as microfiltration. Microfiltration is a means for separating suspended solids from a liquid product of interest. The material which passes through the membrane, now considered clarified broth, is then processed forward. In the fermenter, the acetic acid produced by the organism is neutralized by lime-producing calcium acetate to control pH near neutral. In order to recover the acetic acid, the calcium acetate salt must be converted back to acetic acid, and to do this, sulfuric acid is added in a step called direct acidification. This process is simple and easy to control. After the quick chemical reaction, a dilute acetic acid stream containing some solids is ready for further processing. A belt filter is ZeaChem's current method of choice for the separation of solids from the dilute acetic acid stream. It is a continuous filtration system where the dilute acetic acid passes through while the solids form a cake on the moving belt. The dilute acetic acid stream then passes through an ultrafiltration system for additional liquid-solid separation. The final product produced from the ZeaChem pilot facility is 6% dilute acetic acid. The dilute acetic acid was sent to various downstream vendors for additional process conversion. The vendors successfully concentrated the acetic acid, converted the acetic acid into ethyl acetate, and finally into fuel-grade ethanol. With pilot work on the C2 product platform complete, ZeaChem's 250,000-gallon per year demonstration integrated biorefinery in Boardman, Oregon, will begin operations by the end of 2011. Fermentation systems at the demonstration facility will be 10 times larger than the pilot scale. The pilot facility equipment will now be used to scale up additional product platforms for the new ZeaChem bio-based chemicals and advanced biofuels.